Welcome to the deep dive. We're here to unpack the critical, cutting edge research that helps you stay informed fast. For medical geneticists like yourselves, well, you know the landscape of genomic diagnostics is always shifting. There's this constant demand for methods that are, high throughput, cost effective, and just plain robust. So today we're taking a deep dive into low pass whole genome sequencing, LPWGS. It's a really significant innovation and it's truly transforming how we approach copy number variant analysis.
That's exactly right. Our mission today really is to unpack its current clinical utility, look at the challenges that still exist, and explore some of the truly cutting-edge applications of LPWGS. We'll be drawing on recent surveys, some groundbreaking research, focusing particularly on its diagnostic and prognostic potential in a variety of clinical settings. And that includes its growing adoption, critical aspects like quality control, and its really promising role in liquid biopsy, especially in oncology.
Okay, so let's start with the foundations. LPWGS and its impact on copy number variant analysis. As geneticists, you're all deeply familiar with CNVs, copy number variants. I mean, they're links to disorders like DuGeorge syndrome, that deletion on 22q11.2, or Charcot-Marie-Tooth type 1A with the PMP22 duplication. They're fundamental. And LPWGS is changing how we find them.
Right. Historically, chromosomal microarray analysis, CMA, that's been the first-tier test. But LPWGS, it offers some pretty compelling advantages. You get better genome-wide coverage, which means higher resolution for detecting CNVs across the whole genome, and importantly, higher sensitivity for low-level mosaicism.
Okay.
And then there are the operational benefits. Higher throughput, it's much more feasible to automate, you need less DNA to start with, and lower experimental failure rates. Plus, it's efficient. It uses the same sequencing platform as other NGS tests, like NIPT, non-invasive prenatal testing. So it fits neatly into existing lab workflows.
So CMA was the gold standard. What are the key technical differences then that make LPWGS such an evolution? I understand it's typically under one-fold coverage, focusing on CNVs around 50 to 100 kilobases. How's that different from, say, the high-read depth WGS we sometimes see, like 30-fold coverage?
Yeah, that's a crucial distinction. High read depth WGS, it's aiming for everything, SNVs, indels, the works. LPWGS, on the other hand, is really tuned specifically for CNV analysis. This focus means, well, less manpower needed both in the wet lab and for data analysis. You get much higher throughput and a faster turnaround time, which, makes it pretty ideal for diagnostic labs facing increasing demands and, frankly, budget pressures.
You mentioned real-world application. I know that Zhang et al. Survey from 2022 has some really interesting insights. Before we get into the specifics, what were the expectations, or maybe even some resistance, around LPWGS adoption that the survey aimed to clarify? Especially since a Chinese expert group had already recommended it back in 2019 for certain high-risk pregnancies and other situations.
That's a great question. The recommendation was there, yes, but actually translating that into consistent clinical practice across different hospitals.
Right.
That's where the hurdles often are. This survey, it covered 24 accredited Chinese tertiary hospitals already using LPWGS routinely. After excluding the NIPT-only places, 21 hospitals gave data on their CNV analysis work, and the sheer volume was, well, striking, an estimated annual sample volume over 36,000 cases.
Wow, 36,000.
Yeah. High-risk pregnancies were about 42%, spontaneous abortions around 34%, couples with adverse pregnancy history nearly 20%, and then children with congenital birth defects making up about 4.6%. The demand is clearly huge.
That volume certainly speaks to adoption. But what about the technical side across these different places? Was there much variability in how they were actually doing LPWGS, like detection limits, sequencing methods?
Oh, absolutely. The technical diversity was a key finding. The most common resolution reported was 100 kilobases, yes, but the range was surprisingly wide. It went from 50 kilobases all the way up to 4 megabases.
4 megabases.
4 megabases. And that's not just a technical detail, right? It really highlights a challenge for consistent quality. How do you compare results if the resolution is so different? It directly impacts clinical utility. As for methods, about 62% use single-end sequencing, nearly 29% use paired-end, which we know can improve CNV calling, and a few use both.
And the sequencing parameters themselves, read counts, depth, did that vary a lot too, even for the same target resolution?
It did. The mean read count was around 13.75 million reads per case, median read length 65 base pairs. But to your point about variability, for that common 100 kilobyte resolution, The average depth coverage ranged anywhere from 0.03x up to 1.8x.
Hmm, 0.03 to 1.88. That's quite a spread.
It really is. It underscores these inconsistencies in practice that, well, directly hit quality control and make standardization difficult.
Which brings us right to that point, QC and standardization, especially reporting. How are these hospitals doing with following guidelines like ISCN 2016 and HGVS? What were the common slip-ups?
Yeah, the compliance assessment showed there's definitely room for improvement. The average score for reporting nomenclature compliance was only 7.79 out of 10. Common issues were things like missing the sequencing technology symbol, not stating the genome build, forgetting the G prefix for reference sequences, or not explicitly saying del or dupe for the variant type. And get this, only one hospital actually got a perfect score.
It's just one.
Just one. And this isn't just about ticking boxes on guidelines. It's about the reliability, the comparability results between labs. That's fundamental for clinical use and research sharing.
And what about how the labs were actually set up? Did the survey look at operational patterns and how that affects quality assurance?
It did. The models varied a lot. Some did everything in-house, wet lab and bioinformatics. Others outsourced parts or even the whole process. This kind of fragmentation, it really complicates things like setting up robust external quality assessment programs, EQA, increases risks of sample mix-ups, gaps in QC checks, problems with data sharing. It just makes standardization incredibly tough when there aren't detailed international or even national guidelines specifically for LPWGS and CNV detection, covering everything from wet lab to dry lab to reporting. The recommendation to move to ISCN 2020 helps with reporting standardization, but we still need those comprehensive procedural guidelines.
Okay, so moving beyond the established diagnostics, where is LPWGS really pushing the envelope now? One area that seems incredibly exciting is liquid biopsies, particularly for oncology. For geneticists working with cancer patients, this feels like a potential game changer, doesn't it?
Oh, absolutely. The rise of liquid biopsy is, well, transformative. It's
Right.
minimally invasive compared to tissue biopsies, gives you real-time insights into the tumor's genetics, its heterogeneity, all of which is crucial for prognosis and tracking treatment response. And the key there is circulating tumor DNA, ctDNA, those fragments from cancer cells floating in the bloodstream. And this is where ultra-low-pass WGS or ULP-WGS comes into play. It's a subset of LP-WGS, usually even lower coverage, maybe 0.1x, sometimes even under 0.5x in some studies.
ULP-WGS. That sounds like it really boosts accessibility. What makes it so much cheaper and less computationally heavy than standard WGS? And what does that mean for using it at scale, focusing on CNVs?
Right, the main advantage is definitely the lower cost and computational demand compared to, say, 30x WGS. That makes ULP-WGS really well suited for large-scale screening. And for detecting those major genomic changes like CNVs, it gives you a broad genomic picture, less detailed maybe, but broad, allowing a cost-effective way to gauge tumor burden and genomic instability.
There was that 2025 meta-analysis by Sogby and colleagues highlighting the prognostic value of ULP-WGS for actDNA in solid tumors. What were the key takeaways from that review?
This is a powerful study. The meta-analysis showed pretty clearly that patients with positive ctDNA at baseline, defined as a tumor fraction of 10% or more, using tools like ICR-CNA, had a significantly higher risk of death. The hazard ratio was 2.60 for death and 2.28 for disease progression. And this held true across different cancer types: metastatic non-small cell lung cancer, localized osteosarcoma, metastatic castration-resistant prostate cancer, advanced hepatocellular carcinoma.
That's a really strong prognostic signal. Can you tell us a bit more about the technical specs in those studies?
Yeah.
Coverage, input DNA, and how ULP-WGS really helps analyze these CNAs as markers.
Sure. Typically, these studies used a median coverage around 0.3x, with cfDNA inputs anywhere from 1 to 50 nanograms. The detection rate for ctDNA varied, maybe 28% to 61%. ULP-WGS is just great for CNA analysis. It revealed important markers like losses of 5q and 16q linked to shorter survival in HCC, or in MCRPC, loss of 8p and gain of 9q tied to worse survival. Gain at 8q in osteosarcoma trending towards worse outcomes too. The advantages are clear: easy processing, low cost, quick results, and this ability to map large genomic changes, calculate tumor fraction, and even get a sense of tumor heterogeneity.
So it sounds incredibly powerful for prognosis, but you mentioned detection rates varied. It's not perfect, especially maybe for early detection. Is it more for monitoring, or are people trying to boost that sensitivity?
You've hit on a key limitation, yeah. ULP-WGS does struggle a bit with sensitivity in patients with low tumor burden or very early stage disease, and it won't pick up specific single nucleotide variants. There's also some variability in how samples are handled before analysis, the pre-analytical stuff that needs more standardization. But yes, researchers are actively working on boosting sensitivity, optimizing DNA extraction, improving the bioinformatics to find smaller or more fragmented CNAs.
Okay, so beyond prognosis, LPWGS is also showing value in really tough clinical areas like pediatric CNS tumors. That O'Halloran study from 2023 highlighted the need for minimally invasive options there. Why is this area so difficult, and why CSF?
Pediatric CNS tumors. They're tragically the most common solid tumors in kids and the leading cause of cancer death. The challenge is just how invasive and risky traditional brain biopsies are for these young patients. Cerebrospinal fluid, CSF, turns out to yield the highest fraction of ctDNA from brain tumors compared to blood plasma, so the much better, safer source for liquid biopsy here. That study used a really neat integrated platform, LPWGS, average depth 4x for the genome-wide CNAs, combined with a custom capture panel much deeper at 331x for specific mutations and fusions.
And how well did this combined approach work? What were the detection rates, especially for the CNAs found by LPWGS, and how did that match up with standard methods like CMA?
The results were really quite promising. Overall, they detected abnormalities, CNAs, sequence variants, fusions in 51% of the tumor patients. LPWGS specifically found CNAs in about 24.4% of cases, and importantly, these findings were highly concordant with chromosomal microarray results. Sequence variants had about 60% concordance, and key fusions like KIA1549 by BRA showed 70% concordance. Pretty strong validation.
That is strong. Can you give us a concrete example, a case where this actually made a difference for a patient?
Yes, and this is where it gets really impactful for clinicians, right?
Yeah.
There was a compelling case of an 11-year-old with medulloblastoma. At the end of therapy, they did LPWGS on the CSF-CF DNA. It revealed persistent CNV's gain of chromosome 7, loss of chromosome 8. This strongly suggested residual disease even when other signs might have been absent. And that guided the critical decision to continue with metronomic therapy, potentially preventing a relapse, a decision that might have been missed otherwise.
That's incredible. But are there still challenges? Especially maybe with lower grade brain tumors. Does the approach work as well there?
That's a fair point.
Uh-huh.
Detecting cfDNA in low-grade gliomas, for instance, has been harder. The amount of cfDNA shed seems to correlate with tumor-grade, high-grade tumors shed more.
Hmm.
So yes, while it's promising, especially for high-grade tumors and monitoring, we definitely need more prospective trials. We need to refine how we use these CSF liquid biopsy findings to make treatment decisions and improve sensitivity for those tricky low-grade cases.
Okay, so if that's the current frontier, what's even further out? Pushing the boundaries. Which brings us to single cell LPWGS and this idea of deterministic cancer diagnosis. The big hurdle here is the circulating tumor cells, CTCs, right?
Precisely. CTCs, these intact tumor cells in body fluids, are incredibly valuable because they offer a direct window into the tumor's genome. But analyzing them one by one, that's been tough. You're dealing with low throughput, accuracy issues, and really high costs historically. But this new approach, SCMET-SEC, described by Shen et al. In 2024, looks like a major step forward. Their method combines a metabolic marker, hexokinase 2, HK2, to first find suspicious CTCs, or SCTCs. Then, they use a TN5 transposon-based single-cell LPWGS, pretty low depth, around 0.2x, to profile the CNVs in those single cells.
Interesting. So how do they get to this deterministic definition based on CNAs? Why is that concordance aspect so important for specificity?
That's the really clever part. A positive SCMET-SEC result isn't just finding one cell with CNAs. It requires finding at least two suspicious CTCs that show concordant CNA profiles. The thinking is, CNAs are almost everywhere in solid tumors, but in normal benign tissues, if you see them at all, they're sporadic, random. Requiring concordance between multiple cells drastically increases the specificity. You're much more certain you're looking at true tumor-derived cells.
And just how different is the CNA burden, the amount of altered genome, between cancer and normal cells? Is it a clear enough difference to be a reliable marker?
It seems to be remarkably clear, yes. The CNA burden, the percentage of the autosomal genome that's altered, is reported to be 23 to 90 times higher in cancers compared to normal controls. They found that using a CNA burden threshold of just zero point zero 0.02, sensitivity for detecting various cancers ranged from 55% up to 100%, while the rate in normal controls was only 2.2%. That's a very strong signal.
Okay, that's the science. But practically, how did they make single-cell analysis scalable and affordable with SCMET-SEC? What were the technical leaps?
Yeah, that's crucial for actually using it. Two main breakthroughs. First, they developed a better cell fixation method using click chemistry, which minimizes DNA damage. This apparently boosted sequencing coverage by about 30% compared to standard PFA fixation. Second, their TN5-based protocol is really streamlined. It combines whole genome amplification and library prep in a one step. This cut the processing time way down from maybe 8.5 hours to just 3 hours and slashed costs by something like 97% compared to older methods like Melbake. Big steps for practicality.
So, clinically, how did SCIMET-SEC perform? What about in challenging situations like diagnosing malignant ascites or even metastatic lung cancer from blood?
The results look very promising. For malignant ascites, it showed strong ability to discriminate. SETC counts were much higher in malignant versus benign fluid. If they found 2 or more SETCs per ml, they'd proceed with the single-cell WGS. And the accuracy was impressive. 79% diagnostic sensitivity, but with 100% specificity and 100% positive predictive value across more than 10 cancer types.
How does that 79% sensitivity stack up against standard cytology for malignant ascites?
It's significantly better. Conventional cytology sensitivity is often cited around 52%. Interestingly, they found combining STEC-SEC with cytology pushed sensitivity up to 82% while still keeping that perfect specificity and PPV. And critically, the CNA profiles they got from the CTCs matched the profiles from the patient's actual tumor tissues, confirming their origin.
Okay, beyond ascites fluid, what about blood? Did it work for diagnosing metastatic disease like small cell lung cancer?
Yes, they validated it for that too. Using blood samples, SPECNet-SEC achieved 90% diagnostic sensitivity for metastatic SCLC, again with 100% specificity and 100% PPV. And just like with ascites, the CTC showed identical CNA patterns to the bulk tumor tissue, validating blood as a medium for diagnosing metastasis this way. So the advantages are pretty transformative. Potential for deterministic multi-cancer diagnosis with high accuracy, and this unique capability to diagnose metastasis by finding CTCs in fluids far from the primary site.
It definitely sounds compelling, but as always with new tech, there must be limitations or challenges still, right? What are the remaining hurdles for SCUP-MedSec?
Indeed, there are caveats. A small number of tumors might just lack detectable CNA, so sensitivity could be limited there. The cohort sizes in these initial studies are still relatively small, so we absolutely need larger prospective trials for broader validation. Also, while CNA profiles screen cancer, they don't tell you the exact tissue of origin or the specific cancer type, and the HK2 marker they use might be less sensitive for tumors that aren't highly glycolytic. So, still work to do.
It's really fascinating, though, to trace the journey of LPWGS from, a promising alternative for CNV analysis to this really sophisticated, multifaceted tool in oncology. It's rapidly changing genomic diagnostics, offering higher throughput, lower cost, and increasingly precise insights.
Absolutely. And the common thread, the really unifying theme through all these applications we've discussed, is the power of copy number alterations. CNAs are really emerging as this foundational biomarker for diagnosis, for prognosis, for monitoring disease, regardless of the specific flavor of LPWGS being used.
So, as medical geneticists, you listening are right there at the forefront, translating these advancements into patient care. It makes you think, doesn't it? How might the growing standardization, the automation of LPWGS, especially combined with things like single cell analysis, how might that not just streamline workflows, but maybe fundamentally change how we approach early detection, treatment stratification, even long term surveillance in clinical genetics? And what new ethical questions, what logistical challenges will come up as these incredibly powerful, non invasive genomic insights become even more routine in our practice? Something to consider.